Today, I have the honour of introducing you to Dr. Mike Byrne. And Mike is Clinical Professor of Medicine at UBC, or the University of British Columbia, and also the CEO of Satisfai Health. And we will speak today about using artificial intelligence in gastroenterology. So welcome, Mike. How are you doing?
Good afternoon, Dublin. Good morning from Vancouver.
Well,
I'm
good
well, thank you.
morning. And it's wonderful to have you here, Mike. And first question for you is maybe you could give our audience a brief snapshot of your career and how you got to be CEO of Satisfy Health.
Indeed. So, I go back, well, when you and I were in the lab together in Dublin, I was doing most of my GI training there as a registrar or a fellow, and became a clinical physician in North America for the last 22 years, Duke, and now Vancouver for 20 years, my delightful home. And 8, 9 years ago, I had an itch that I couldn't satisfy, and in terms of doing something different. And I right connections, right networks. A couple of ideas that I had. I put together a consortium to look at one particular need for AI in the GI space, and it's blossomed since there.
Yeah, so that's what I want to ask you about next is why did you you're almost a decade now CEO of Satisfied Health. Why did you start it? What was that itch that you had to scratch?
I wanted to get into entrepreneurship, but it's also that there is a pressing need in medicine, as I'm sure you're very familiar with, increasing human performance. So in my space of GI, a lot of the work that we do is endoscopy. So cameras into the bowel throughout the intestine. And despite the fact that we've had significant technological advances for the last 20 years, better resolution, higher magnification, better lighting, better on-screen detail, human performance has stagnated. So we just simply can't extract all of the information from the data on the screen to make informed decisions. So hence the need for digital technologies. And that really excited me at the time.
So what is the most exciting thing, I suppose, that you have found in those 8 to 10 years? What's the most exciting outcome for you?
I think the overall idea of being able to do what's called an optical biopsy or a virtual pathology. And what that means is I'm doing a live procedure. I have a screen in front of me with the esophagus or the colon, and I see something. And rather than having to take always a physical biopsy and send to the lab and wait for a week or two or what have you, there is an increasing ability for machines to look and in live time, within milliseconds, 20, 30, 40 milliseconds, tell you that this is pathology type A, B, C, D. And that's really transformative in terms of accuracy, in terms of cost saving, in terms of being able to tell the patient there and then what's going on.
So you did this for colon polyps. So maybe you could tell us more about colon polyps, about detecting them, about adenomas and adenoma detection rates. And so tell our audience more about this because it's fascinating.
Colon polyps are, colon screening is very important. The third commonest cancer globally, colon cancer, and alarmingly happening in younger and younger people, as we've seen with some, media celebrities in the last couple of years, unfortunately being diagnosed with colon cancer young. Most colon cancers develop from polyps that are called adenomas. Many of us over the age of 50, myself, many of us have colon polyps that we don't even know about. But some of those may turn into cancer. So we screen people. Okay. There's a metric called the ADR, or the adenoma detection rate. It should be 50, 60, 70% in an ideal scenario, like experts, but many people, it's 20, 30, 40%. And machines have already been shown to improve that by at least 8, 10%. And given that there's a stat that for every 1% increase in the adenoma detection rate, you will decrease colon cancer mortality by 3%. To me, it's unarguable that you should be using technology, physician and machine, in kind of perfect harmony.
So this idea is physician machine together increase the detection rates. So you're saying you got an additional almost 30% decrease in mortality. Would I be right in saying that?
Yeah, I guess if I do the math on that, so yeah, it sounds like a very great ask, or a very broad claim. But the studies have shown over the years that if you simply just move the needle a little bit in adenoma detection, you're having a significant impact on population-based colon cancer.
That's really exciting. And is that in the clinic now? Is Satisfy Health got that in the clinic right now?
So we don't have that in the clinic today. We have solutions in that space, and we've got a broad range of solutions that we're working on to try and put together in a platform approach. But there are commercially available so-called CADI, or computer-aided detection tools of polyps out there from some big industry players for sure.
Okay. So in terms of Satisfy Health, in terms of your pipeline, then what do you have in your pipeline?
So there's the polyp work that you just talked about. So that's both detection and characterization. In other words, being able to do an optical biopsy. We talked about that. We are doing a lot of what I believe very exciting work in the upper GI tract. So in the esophagus, for example, esophageal cancer is on the rise. There was a paper I read yesterday that esophageal cancer, which was erstwhile felt to be less common in women, is actually increasing significantly more in women than in men for reasons that are unclear. So that is a much more difficult realm for doctors to be accurate about, because it's much more of a field defect, flat often, rather than individual visible polyps. So the idea to move from what we do right now, which is non-targeted biopsies of what's called Barrett's esophagus, which is a precancerous change from reflux, to doing targeted biopsies and going where the machine says, I think there's something unusual or strange there, could be a paradigm shift in terms of how we assess, and also decreasing the significant miss rate that we have in early esophageal cancer.
So you're
Yeah.
saying basically that you will be able to go from this idea of right now non-targeted biopsies to actual targeted biopsies in specific areas in the esophagus, so that you could actually detect cancer earlier. Is that correct?
Absolutely. And
Wow.
not only moving to targeted biopsies, but actually just increasing the yield, because even the multiple non-targeted biopsies often still miss the sweet spot where there's something bad going on.
And so you have, so that's colon polyps, esophageal cancers. Do you also have solutions for IBD as well? Is that correct?
Yeah, I mean, the thrust of what we're doing at Satisfy is to bring better, extract more information from what we do in live endoscopy. So whatever the GI organ is the largest organ in the body, all the bowel and all the digestive organs. So another common condition is Crohn's and colitis, or inflammatory bowel disease. So we're very excited about some of the work we're doing with ulcerative colitis, whereby in clinical trials, if you want somebody to go into a trial, you need to meet the inclusion criteria, as you know. And right now, that's all done with so-called human expert readers who sit and read videos that are recorded of patients to see if they match the criteria. It's timely, it's subjective, there is error, and we've got machines that will do it really quickly in the clinical trial space.
Okay, and then we'll define that this patient is correct for the clinical trial. So tell us, so in terms of what you said about looking for more information from your screening, from basically bowel screening, what do you mean by that?
So if I go to the colitis, getting more information, right now the scoring system, I'll make it easy, is what's called the Mayo score. 0, 1, 2, 3. 0 meaning no disease activity, 3 meaning very inflamed, 1 and 2 in between. That's a human-driven, solid categories of score. It's a continuum. It's not this categorization. So there are patients who are a 1.378 or whatever. Of course, we don't say that, but the machine is able to see different gradations. And it's likely that somebody who's closer to a 2 than a 1, which the human eye can't differentiate, are different patients, and their biology is different, and their response to treatment is different. So the ability to get more granular in what we're seeing on the screen, rather than this fairly blanket assignment of disease activity, and potentially, looking, which is one area that we're focusing on, for image-based biomarkers that are predictive of response to treatment, or cancer recurrence in the future or whatever is really enticing. And we're just at the beginning of that journey in the image space for endoscopy.
So I think this is fascinating. So basically, are you saying that as you're capturing the image, that you can basically capture, using the AI, capture thousands of features from that image, as opposed to going back to what you said, the Mayo score, which is one, two, three or four different markers from that image, but you're basically speaking about capturing thousands of features and
Yeah.
and which are the important variables, and connecting that to the whole picture, if you like, of the full colon.
Yeah, absolutely. So if we go back to polyps again, to do this human optical biopsy, we are trained to look for three features: color, pit pattern, and vascular pattern. That's it, three features. The machine, some of the work that we've done, for example, if you tease the network apart, you see that it's looking at least 1,000 features per polyp, things that we can't even perceive as humans or understand. Lots of zeros and ones, edges, you name it, you understand it. So there are some things where we just have to put our hands up and say, you know what, we are limited by what we can see, and fatigue and what have you. So why not let a machine help us?
And that's kind of rolls into my next question about pushback. You're saying people, people may not like to use this, or they don't see the value in it. But speaking to you now, it's pretty obvious what the value is. So I suppose, how do you respond to people who say, there's no need for this?
So it's very important to get physician adoption and patient comfort, right, with AI technology. Patients might say, I don't want a machine telling me if I've got cancer or not. I want the doctor to do so. But if they are aware that there's a significant miss rate in my space of GI in live procedures, significant, and that machines can improve that, I don't see what the argument is. It's a pretty yes and no argument to me. So, for example, again, back to polyps. If my polyp detection rate is 30%, that's low. And there are plenty of people operating at that level out there, plenty of them. If I can increase their performance by 5, 6, 7, 8%, that's already a significant improvement. And if you do that across the whole space and all the physicians, the savings are incredible. So anybody who pushes back when they're a low performer, which is many people, that's my very simple argument back to them. And there are many, many, many more beyond that. But that's a pretty easy metric to say to them and have them give you a valid answer.
And back to your save lives, your 3% increase in mortality, decrease in mortality. So it's a pretty much a no-brainer. But in terms of, I suppose, cost of this, you'll have a lot of people arguing around costs and reimbursement costs for patients. What is Satisfai Health? I mean, what's your thoughts in this area?
Yeah, cost implications are huge, and it's one of the barriers, actually, Patricia, to adoption of AI technology in medicine, because we haven't yet figured out, certainly in the GI space, it's starting to move in the radiology space. We haven't figured out who's paying for what. If there's no reimbursement for AI tools, it's going to be very hard to get industry traction and adoption in the market. So that needs to change, and fee code bodies or health organizations or the National Health Service or the Health Service in Ireland has to look at the overall potential savings here and recognize that it's going to improve care or it may even save cost. It makes decisions more objective. I'll give you a quick example on, let's say, colitis. So I talked about the central reading aspect. In clinical practice, if I see a patient with colitis and I'm doing a procedure and I want to maybe change their biologic, their expensive drug, I'm doing it based on what I see usually on the screen. It's quite subjective. If I'm a third-party payer, I want to know that you use the most accurate assessment in live time, and that may be where bodies, reimbursement bodies, insurance companies, whatever, that may be where they go and say, show me the most accurate metric that is out there, measurement, and then I will cover your drug change or your drug continuation or what have you.
Yeah, it makes so much sense. And unfortunately, we're running out of time, but I have one more question for you. And I'm really, really looking forward to hearing your answer to this. You're in the field of AI in healthcare, and there's lots of issues, there's lots of barriers. But what I suppose is what excites you about the future? You know, what do you see as the most exciting future development around AI in healthcare?
So it's literally that we're just at the beginning of this journey. You know, there's been so much hype, whether it's medical AI or non-medical AI for the last several years, and in the medical space where we have to be much more careful because of potential harm to patients, etc. We're just starting. It really is at the beginning of a very exciting journey. I have no doubt that this old adage that I've seen on many slides for the last couple of years, which is AI won't replace physicians, but physicians who use AI will replace physicians who don't. I truly believe in that. So I'm excited because I think we're really just scraping the surface right now.
And we wish you so much the best for your future and for the future of Satisfy Health. It's just so intriguing, and we are completely out of time. But sincere apologies to all the audience whose questions I did not get to. But I just wanted to take this opportunity to say a huge thanks to Mike. It's so great to see you again, Mike. Thank you so much.
My pleasure. Thank you very much for the invitation.
Yeah, it's our pleasure, and thanks for everybody for joining in. And please do check out our website for our list of upcoming speakers, and see you all soon at our next HIT series. Thank you so much. Take care. Bye.